The purpose of this video is to introduce a new type of Punnett square problem. They're called dihybrid crosses. In this case, the prefix di tells us it's using two, and it's two traits that we're looking at. So everything we've done previously, we've just looked at one trait for the individual species. So we've looked at things like, flower position, if it's axial or terminal, or flower color, things like that. Now it's getting a little bit different. Now we're going to look at two traits for the same individual. So for example, if we're sticking with the Mendelian pea plants, maybe we would look at the plant's height as well as the plant's flower color. So you can look at more than one trait when you're doing dihybrid crosses. So just a couple of notes for these guys. The first thing is we're looking at two traits here. The other thing is this is why we're using the capital and lowercase letters. Because at this point, since we have two traits, it would be very confusing if you were using, one letter for dominant and another letter for recessive. So this is why we keep it as the same letter. It's the first letter of the dominant trait. We just have it be capital for dominant, lowercase for recessive. These Punnett squares are a little bit more complicated. These are going to be 16-box Punnett squares. What that means is that it's now going to be a little bit more complex for you to set up the problem. Before, it was very easy to take the parents and put them in the Punnett square. I mean, you just put, one parent along the top and then the other parent down the side, whatever it is that their genotype might be. Now in this case, it's not going to be quite that simple. There's going to be a process that we'll go through to talk about to figure out how you get what goes on the top and what goes on the side. It's not going to be as easy as just taking the genotypes and placing them in there. So I'm going to spend most of the time in this video just going through an example problem with you in order to give you an idea of how to better answer these types of problems. So to start this problem, we'll begin the same place we always do, just kind of reading through it and getting the information out. It says two plants are bred. The first is homozygous dominant for orange flowers and heterozygous for being tall. The second is homozygous recessive with white flowers and homozygous recessive for being short. So one of the first things to do is just to go through and write down what is dominant and what's recessive, like we've started with so many times. Again, I don't give you a space for this necessarily, but this is still an important part of the problem. So to start off, got to figure out what the dominant traits are. For starters, if we're looking up here, we'll see that it says one is homozygous dominant for orange flowers. So we know that orange is one of our dominant traits. So then we're trying to figure out what the recessive traits are. The flip side of that one is white. Now the other trait that it talks about is the height of the plant. So if we look back up here at our problem, it says this one is heterozygous for being tall. Remember, the heterozygous individual has one dominant allele and one recessive allele. So the heterozygous one will always show the dominant trait. It will always have the dominant phenotype. Because of this, we know that tall is the dominant trait. Then the flip side of that one would be short. So this gives us an idea of what we're using for our letters. The next thing we have to do is figure out the parent genotype. So if we look at our first individual, it says it's homozygous dominant for orange flowers. Since we know that orange is our dominant trait, we're going to use O's. Since it's homozygous dominant, it will be two capital O's. So capital, capital. Since my O's all kind of end up looking the same, I'm going to write the lowercase o sort of as like a cursive O to keep me from having to go through and underlining everything. For the next part of this one, it says it's also heterozygous for being tall. So we're actually going to write down 4 different letters for this parent. Since it's heterozygous for being tall, we know the prefix hetero tells us it's different. That means it has one dominant allele and one recessive allele. So the end of this one's genotype is capital T, lowercase t. So now we need our second parent. It says the second parent is homozygous recessive with white flowers. Since white is a recessive trait, the only way to have that is 2 lowercase o's. And then it also says it's homozygous recessive for being short. So that means it'll have 2 lowercase t's as well. So now instead of just having 2 traits for a genotype, we're actually going to end up having 3 separate traits. I'm sorry, instead of having one trait for a genotype, so which would give us 2 alleles, we'll now have 2 separate traits, which ends up giving us 4 alleles. The tricky part here has to do with how you take this and then fill out the top of the Punnett square. What I see people do a lot of times is they say, oh, this is easy. I have 4 different alleles here, so I just put those 4 alleles along the top of the Punnett square. That's not accurate, because if you're looking at our parents here, we end up with 4 alleles in each individual. So you actually need to end up with 4 letters in each box. If you do this and put 4 across the top and 4 across the side, you only end up having like a much smaller number. You'll end up with just 2 in each box. So we'll get rid of that and go about this a different way. There's 2 ways of figuring out the letters that you'll put up there, and I'll show you both of them along the bottom here. The first way is to take our parent's genotype and make a small 4-box Punnett square. So we're going to take the first part of it, the homozygous dominant part, and we're going to put that along the top. We're going to take the heterozygous part and put it along the side. Then what you end up doing is combining all of them together inside the box. I'm going to write the letter O first just because I did that up at the top. It really doesn't matter. What does matter is that you stay consistent with it. So even though it doesn't really matter which one you pick. So that's one way of doing this. You can make the small box Punnett square. These then become the 4 things that we are putting in the chart up here at the top. So we would write capital O, capital T, capital O, capital T, capital O, lowercase t, capital O, lowercase t. The other way to get that is to FOIL in order to get it, if you know FOIL from math class. So if we're thinking of the whole FOIL concept, FOIL stands for first, outside, inside, and last. So in order to FOIL, you would combine your first terms together. And I guess an easy way to think about this is to sort of put it in parentheses like that. So our first term over here would be capital O, capital T. So we'll write that one down. Our outside terms would be the ones here and here. So that would be capital O, lowercase t. Now we have our inside terms. That would be our capital O and capital T again. And then our last terms. So that would be our capital O here and then our lowercase t. So if you notice, I'll just write this one down at the bottom here, that gives us the exact same thing as what we got in the Punnett square. Some people find FOIL to be faster. I think this is much easier. If you're not necessarily someone who excels in math, I would suggest just making the small box Punnett square. If you're concerned about doing these problems quickly and you're comfortable with the FOIL method, then you can use that one. It truly makes no difference to me because you end up with the same answer, no matter which way you do it. Now, if you take the time to do one for this one, you'll see this one comes out quite simply because we have 2 recessive alleles for both traits, so they'll actually all be the same thing. They'll all be lowercase o, lowercase t. If you doubt me, you can always go through and check it on your own. Just to kind of speed this up for time, if you want to, you can take my word for it. We'll go through and fill those in. Sorry that my lowercase t's, my cursive t's, get a little sloppy. Now this is where it's going to get tricky, trying to write all this stuff in that box. What I'm going to do, just to make my life a little bit easier, is I'm going to blow up this image a little bit for us. So now, just to go through and combine these together, we've got capital O, lowercase o, capital T, lowercase t. And actually, this is going to be the same for all of the columns, because this left-hand side, since these are all the same, every column for this particular problem is going to be the same. That will not always be the case, but that's going to kind of speed things up as we're putting this together. So now for this one, we end up with the same thing actually. Big O, big T, little t. Now in this column, it's a little bit different. We've got our big O, and then little o, and then 2 little t's. And that'll be the same all the way down. Oh, look at me, I'm putting the tail on my capital O. And then it'll be the same thing in the last column. Big O, little t, little t. Alright, so we'll shrink this one back down again. Now that I'm finished writing in there. The next step, and I know this is getting a little long, but these problems are just a little bit involved. The next step is the offspring phenotypes. So what do the offspring look like? If we take a look at our column over here, we'll just take this one column at a time since all of the columns happen to be the same. So this one, if we just look at color, this would be orange and tall because the dominant allele for O and the dominant allele for T take over. So we'll write that in here. Orange plus tall as one of them. Now that happens to be the same as the ones in this column. These guys, though, are orange and short, and it actually ends up being the same in the last column. Now again, keep in mind, the only reason all of our columns are the same here is because all of these traits are the same. If you end up with a mix on this side, like we have a mix up here at the top, then you'd end up with different things in the column on the inside. But for now, it's a little bit easier, so we're starting off with a simpler problem. So our other option is orange and short. The last thing is the phenotype ratio, which is like the odds of getting each one. For this one, it's a 50-50 ratio because we have half of the Punnett square answers, half of the columns. That's capital O, capital T, showing us that this is orange and tall, and then half of them are orange and short. So there's two ways to fill this one out. You could call that a 1-to-1 ratio, or you could just say that it is 50-50. It really doesn't make a difference to me that those are two ways of representing the same concept for our outcome from over here. Some of the ratios will get a little bit more complicated because we end up with like a wider variety of traits. So I honestly think that this is probably your better option to go with rather than thinking of it as 50-50. Another way to do this, just to show how we got 1-to-1, if you were to count all of them up, all of the boxes that are heterozygous for both traits, there's 8 of them. So if we count them, just sort of check them off, we got 1, 2, 3, 4, 5, 6, 7, 8. So 8. And then if we go through on this side, all of the boxes that are orange and short, we have 8 of those as well. So 1, 2, 3, 4, 5, 6, 7, 8. That started off as an 8-to-8 ratio, which then we reduced down to 1-to-1. Just so you understand how we got that, like where that ratio came from. I really think that's easier than doing this, although both of them are technically correct. Another thing I suggest you do, as I did, is to check off those boxes as you go through there. That way you're sure you didn't miss anything. And keep in mind that whatever numbers you have in your ratio here, they should always add up to 16 because there are 16 boxes in our Punnett square. So I know this one got a little bit long, but I appreciate you guys watching. This is a little bit more of a complicated problem. To be honest, my opinion on these is that they're not really difficult. They're just kind of involved, and they just take some time. Once you do a few of them, I think you'll become comfortable. I honestly think the non-Mendelian ones are probably harder than this, even though they don't take quite as much time to complete. So again, I know this video got a little bit longer. I appreciate you hanging around and watching. Take a minute to answer the questions at the end, and we'll practice some of these in class. Thanks.